Dear students and judges, we're the phosphate team and we are here to share our project on a biosensor. We will be discussing our topic based on the background, experience, and human practice. Our logo consists of two main subjects. The fire on the upper part represents phosphate because of its inflammability, and the down part is a hand-shaped water, which symbolizes our hope to control the phosphate pollution. Our name phosphate came from our main focus on the polyatomic ion phosphate. We believe with our project, we are able to control the fate of phosphate and reduce pollution in water in order for all of us to embrace the bright future. For our project, we create a biosensor that can quickly detect phosphate concentration in the water. Our team Phosphate has 21 team members from all over the country of China, and for me personally, it has been a wonderful journey to participate in hygiene with my team members.
Phosphorus is one of the most important elements plants need to survive. Phosphorus can be used to modulate processes of metabolism and development in plants. By forming T6P, it plays a central role in regulating carbohydrate metabolism. As a result, phosphate fertilizers were widely used by farmers all over the world. However, too much phosphate will do harm to the environment, and the most common one is eutrophication. Eutrophication results from the overly released nutrients and minerals for which the growth of algae become excessive, and it may cause the deterioration of water quality and the demise of aquatic organisms. Since phosphate is critical to the growth of algae, the release of excessive phosphate will intensify eutrophication. The widely used phosphate fertilizers and laundry detergents are among the largest producers of phosphate waste, along with industrial waste. Nevertheless, though industrial waste is the most detrimental one, it has developed process of sewage treatment. The domestic waste and agricultural sewage, however, lack sufficient methods of waste collection, so the problem of non-industrial waste is much more complex. Although China is facing a serious problem of sewage treatment, the society and factories do not fully understand that the most important and efficient way of controlling pollution is to limit the discharge of pollutants, especially phosphate when we are talking about water pollution. Thus, we aim to raise the public's attention to water pollution control and design our product environmentally friendly to match our value. Our goal follows iGEM's ideology of local people solving local problems. We found that people around our community are all worried about water quality, but they have limited knowledge about the formation of sewage and efficient ways to reduce the discharge of sewage. Taking advantage of the data era, we decided to raise the public attention mainly through social media platforms by uploading visually appealing posts as well as some handy tips. On the other hand, using synthetic biology, we design a product in the form of a biosensor to quickly and qualitatively detect the phosphate concentration in sewage. It is relatively cheaper than what the current market offers, so that river monitoring stations and fishery-related management authorities can detect water quality with lower costs.
I'm going to present the methodology part. To put it at the first place, everything about our experiment is based on the fact that PHOA can be suppressed by phosphate. But at how much extent? That's what we did in our experiment. To find out more detailed information about PHOA and put it into functional use. We want to build a cell-based biosensor using recombinant DNA technology that can help us detect whether there is excessive phosphate in water or not. Using PHOA as the sensor gene, we add a reporting gene GFP to the end of the PHOA sequence so that we can detect its expression condition by the fluorescence intensity by creating the sensing and reporter modules. Analyzing the sequence of PHOA gene, we found out the minimum length of valid promoters and the extending place where we would connect the GFP gene. In principle, our experiment follows the methods of controlling variables. To verify the initial expectation of the fact that PHOA will be suppressed by phosphate, we need to set up a control group that doesn't contain PHOA. Therefore, we inserted only the GFP gene into our vectors part 57 in the control group. We have designed parameters for GFP genes and restriction sites of the equal R1 front and BAM H1 reverse by using parameter 5 so that we can connect the GFP genes with the vectors in the way of restriction enzyme digestion. It is important to note that the control group also contains the promoter of PHOA gene in order to make a strict comparison with our experiment group. The verification process of the control group, which contain no PHOA gene, and the experiment group contain PHOA gene, involves treating the two groups with different concentrations of phosphate while observing the change in fluorescence in different time points, which will provide evidence that our indicators work. Other conditions such as temperatures remain the same. We expect that the control group will present a stable amount of fluorescence or totally no fluorescence no matter what concentration of phosphate there is, while the experiment group will show a constant change in fluorescence due to the change in phosphate concentration.
For the process of our experiment, we mainly import the EGFP gene into the vector to construct the control group bacteria. Then we put both the experimental group and the control group separately into MOPS medium, which is used to simulate the environment of wastewater and finally detect its fluorescence. For the specific procedures, we will first do the plasmid extraction. Through these procedures, we obtain our vector PUC57, and then we'll amplify the EGFP genes through PCR process. The PCR results match the expected EGFP length of around 1,500 BP. And then we'll use EcoRI and the BAM high restriction enzyme to digest the plasmid and the EGFP gene. Then we connected EGFP gene into the plasmids in the proportion of 3:1 by using T4 DNA ligase. We have designed corresponding front and reverse primers for the EGFP gene to ensure that they can be connected. And then we'll transform the constructed products into E. coli competent cells. In order to verify whether the transformation is correct, plasmid was extracted from the cultured E. coli, and restriction endonuclease experiments were conducted and using electrophoresis to verify it again. This is the result after the DNA digestion experiment. We can clearly see the two bands representing for the EGFP and the vector, which proves that our connection went on successfully. Also, we have sent the products to the companies to test its sequence. Then we will produce the MOPS medium to simulate the wastewater. These are the ingredients. And then we'll put the E. coli contain PHOA EGFP and the E. coli contain only GFP into the medium and allow them to grow and react, which gives the final verification. In this study, 3 initial concentrations of 0 millimole, 0.2 millimole, and 1 millimole were set to preliminarily predict the detection range of phosphate concentration of PHOA GFP E. coli. First, fusion carrier bacteria that contain both the experimental group and the control group was selected and added into fresh MOPS medium, and epicillin was added respectively for the overnight culture at about 15 hours. And secondly, after the overnight culture, we distribute the bacteria into 3 fresh MOPS medium that contain respectively 0 millimole, 0.2 millimole, and 1 millimole phosphate solution, and then make the initial microbiome OD600 at about 0.02. Then the bacteria will be cultured and selected at different time points in 15 minutes, 1 hour, 3 hours, 5 hours, and 7 hours time. Then we suspended them with 10 millimole phosphate buffer PBS, which has the pH of say 7.4, and then the resuspension was diluted for 10 times, which can show the OD600 value approximately the same as the initial OD600 value. And the fluorescence value of the dilutant and the OD600 value were measured respectively, which are our final results.
For our results, we mainly have 2 outcomes. First, we found the detectable range of phosphate concentration of PHOA EGFP E. coli. In this experiment, 3 initial concentrations of phosphate, 0, 0.2, and 1 millimole, were set to preliminarily predict the detection range of PHOA EGFP E. coli when encountered different phosphate concentration. When different concentrations were added to the culture media for treatment, after 15 minutes, comparing with the control group, the E. coli containing PHOA GFP plasmid had a higher detectable fluorescing value in the 0 to 0.2 millimole of phosphate concentration, which was a big difference. This result not only verifies that PHOA protein expression in E. coli is indeed induced by phosphorus starvation, but also provides a basis for us to further set PHOA GFP phosphate detection range in more detail. Second, we found the appropriate phosphate detection time for PHOA GFP E. coli. As the previous test showed significant differences at 0 to 0.2 millimole, we set up the E. coli containing PHOA GFP plasmid and the CK group containing only GFP E. coli in the culture with phosphate concentration at 0 to 0.2 millimole, then sampling at different time periods. We took time to deal with different PHOA GFP E. coli and compared with CK GFP fluorescence detection of E. coli. The results showed that with the passage of time, the GFP fluorescence signal intensity differences decreases. This suggests that PHOA GFP E. coli can detect phosphorus deficiency signals in a short period of time, thus providing the possibility for us to use it to rapidly detect the presence of phosphorus pollution in water.
There are 6 main advantages of our project, namely reusable material, strong specificity, quick detection, high accuracy, easy operation, and low cost. Firstly, using immobilized biological Escherichia coli as a carrier does not require expensive reagents and can be used repeatedly, which overcomes the disadvantage of high cost of physical and chemical analysis reagents and complicated chemical analysis in the past. Secondly, it only reacts to specific phosphate, and it is not affected by color and turbidity. Thirdly, the analysis speed is fast, and the result can be obtained in only a few minutes or 10 minutes. Fourthly, the relative error can reach only 1%. Fifthly, the operating system is relatively simple and easy to realize batch analysis. Last but not least, in continuous use, only a few cents of RMB is required for each case.
In the perspective of results, I believe that a more precise quantitative relationship between fluorescence intensities and phosphate concentration is needed, considering the fact that our curve is comparatively rough now. This involves extracting the RNA of our indicators and carry out the real-time qPCR. Though the housekeeping gene and target genes primers have been designed, we haven't really carried out that experiment. I hope further experiments will contain the qPCR test. Detailed improvements like the activity of E. coli and how to ensure the normal growth of E. coli without affecting the life activities of other microorganisms in the relevant growth environment. That is, how to protect the large intestine. The safety of phosphorus receptors needs to be considered. Besides, we will also face many unknown challenges, such as how to control the growth and reproduction of E. coli, how to make our phosphorus receptors more intelligent and diversified, and how to reduce the cost of cultivating E. coli and producing phosphorus receptors, how to improve the detection sensitivity of this sensor. These are all the premises for marketization and commercialization of our phosphorus sensors. In perspective of products, a kit still has disadvantages since it can only be limited in the lab. Our product's broad perspective grounds on the real-time on-the-scene detection, which means the microplate system will be replaced by portable GFP detectors. A transition from quantitative detection to qualitative detection may be needed. To be specific, a limit of phosphate concentration is set, and if the sample exceeds the limit, no light will send out. Detailed problems also need to be considered, such as how to preserve the bacteria we constructed and ensure its biological activities, so that the fluorescence will only be affected by phosphate concentrations. Temperatures vary from place to place and may cast uncertainty to our results. Actually, our indicators have this potential to be used in real-time on-the-scene detection of natural water resources because their sensitivity phosphate concentrations are around 0 millimole to 0.2 millimole, which is close to the phosphate concentration standard for lakes or rivers set by the government.
Integrated human practice. Our products aim at minimizing the pollution to the environment while enabling our customers who need to perform field study to have a direct and immediate understanding of the water quality of the tested sample. Through biological means, our customers who need to control the water quality can further decrease their costs in monitoring the phosphate concentration in the water body. In order to achieve this goal, we interviewed experts in related fields and collected some useful advice and suggestions to improve our products and features better. About education and public engagement, we, Team Phosphate, introduced synthetic biology and our project to the public, raising awareness of the adverse effects of excessive phosphate in water. Through these activities, we gathered a group of people interested in science. We inspired more young kids' love of synthetic biology through simple experiment and easy understanding explanation of biosensor and GFP, green fluorescent protein. After all, paying attention to water quality monitoring is not an issue merely to detect bureau, but the more general public. WeChat official accounts. We opened up a WeChat official account called Phosphate to post the daily activities of our team. The official account, as a tool for multi-directional communication, provides us and the public a convenient stage to communicate. Those tweets we post include surveys, interviews with experts, and visit to the manufacturing factory. We combine both professional knowledge and funny language to help public understand some concepts easily. Transforming the elusive ideas into lucid words and pictures, we hope more people can understand it. Through this approach, the audience, targeting friends in social network platforms who may not notice the harm of excessive phosphate in water, can read our tweets and get some recognition for it. Weibo. To introduce the idea of biosynthesis and the control of water pollution to the different social groups within a broader range, we opened up an account called Phosphate SH in Weibo. Weibo is a larger platform, helping us disseminate the information more quickly and broader. We keep updating some photos we took during the experiment and activities to provoke the interest of readers. With interest in this topic, the public can see information we want to popularize. In these articles, we elaborate on the details of the reason why we need to pay attention to the phosphate pollution, how the phosphate pollution influences our daily life, and how we can do to deal with this issue.
After understanding the various types of research methods, we focus on sending a survey and doing interviews. Online reviews can give paper-maker answers pertinently. Also, it can save time and money. That is why we always use it. Online surveys turn out to be our first choice. Through this survey, we gauge the level of residents' knowledge and satisfaction with sewage treatment. Considering the questionnaire form, we set all questions as multiple choice, recognizing that interviewees might give us vague answers if open questions are designed. We sent out 12 questions on water pollution and sewage treatment. The result indicates that nearly 80% of the interviewees feel that it is necessary to government sewage. We noticed that the crowd has a strong demand for sewage treatment while analyzing the collect data. Up to 95% of investigators think that sewage treatment is necessary, but nearly 90% of them don't understand or understand marginally about the consequences of water pollution. As a result, the government should strengthen the management and publicity of water pollution.
We hosted special events in our office building, which was a project presentation. Because every student and their parents were very interested in our topics of the project, we prepared for a long time and decided to present it in front of everybody. We cherished this opportunity to practice and view it as a formal competition, thus getting lots of positive and useful feedback from the audience. The audience was very interested in our unique themes and asked many questions about it. Besides, some professional people gave us valuable suggestions to make improvements. We realized that we still had lots of things to be improved, not only the fluency of our presentation, but also the details of our demonstration. Here, we are grateful for everyone's arrival and listening.